right. Well, welcome to this little discussion that we decided we wanted to share with everybody around the world. We often get the question with Sturge-Weber and from the foundation is like, why do you do what you do? And, what's the impact? Do you have any plans? And so we decided that we were going to invite today Dr. Matthew Shirley, Matt Shirley, and Dr. Jeff Loeb to illuminate some of the questions that you've had and that we get, and also to provide a framework for how we're going to go forward. We'll start with Dr. Shirley this morning, and we appreciate you coming and answering some of these questions. We've gotten a couple that I think you'll be able to handle very ably. One of the ones was, how can cancer research efforts apply to SWS and vascular overgrowth issues?
That's great. And thanks for having us here today, Karen. I think I work in cancer research, and that's what I've been trained in. And I think the question about how we can translate findings from oncology research, it's really relevant because the mutation that causes Sturge-Weber syndrome is also in the same gene, the same mutation that causes uveal melanoma in some people. That's a cancer of the eye. And so, obviously these are two different diseases, but there are some therapeutics developed in the cancer world. One, sirolimus, has actually been used in clinical trials for Sturge-Weber syndrome. And so there are some, immediately obvious drugs that we can try to repurpose. And there have been efforts ongoing for a few years now to see if those are safe for people to take. I think the biggest question that we have when we're trying to use cancer research in treating diseases that are not cancer is, are the therapeutics safe? Because there's obviously for cancer indications, there's a lot more acceptance of sort of toxicity and side effects. So I think that's one main challenge that we might have in Sturge-Weber syndrome. There are good therapies for cancers such as uveal melanoma. However, there are sort of unacceptable side effects when you're giving those drugs to people who have something like Sturge-Weber syndrome. What we can do is try to develop models that we can study that are not actually people, so that we can give the drugs to mice or fish, and we can try to understand safety in those animals first, and try to understand if there are treatments that will work to address the vascular and neurological symptoms of Sturge-Weber syndrome.
I would think in Sturge-Weber syndrome that it's even more of a challenge, because unlike cancer, you're able to deliver those therapeutics directly to the tumor or to the site. And with ours, we kind of have to maybe be a little more aware of the vascular issue. Is it going to take it to the brain when it should stay in the skin? Is that a challenge?
Yeah, so this is a challenge also with cancer therapy. You usually have to try to target a drug directly to the tumor, and that's not always possible. So what we typically try to do is find something called a therapeutic window, where the drug can affect, say, a tumor more than all of your normal bodily organs. And what we haven't defined in Sturge-Weber syndrome is exactly how we can understand that therapeutic window, how we can affect, since Sturge-Weber syndrome is a mosaic syndrome in normal tissues, it becomes much more difficult to try to figure out how to target abnormal vasculature that's right next to normal vasculature. Whereas a tumor can be much more focused and much more localized, and you have things like radiation therapy that are very specific.
Could you speak a little bit about why tissue samples and maybe aqueous humor are so important to our arsenal of tools that we need to battle this?
Yeah. So I think most of the research that's been successful in Sturge-Weber syndrome has stemmed from patient tissue donations, either from skin biopsies of port wine stains or from affected brain tissue from hemispherectomies to control the seizure symptoms. And these tissue donations are critical to support the sort of basic science research, understanding how Sturge-Weber syndrome is and what it is. The work that I did with Dr. Jonathan Pevsner as part of my graduate research to define the genetics of Sturge-Weber syndrome was only made possible because of these patient tissue donations. And several other researchers have made something called cell lines from Sturge-Weber tissue samples. And these are a critical research tool. These cell lines can be used to understand if a treatment is effective or safe without giving that treatment to a person. We also can use these patient tissue samples for a variety of purposes when we're trying to develop things like mice that might mimic Sturge-Weber symptoms, so that we can test these potential therapeutics in mice instead of humans. So really, it's critical that we preserve these precious samples, and that whenever possible, we can accept donations of patient tissue. The aqueous humor is very important too, because we really don't have an understanding of why the Sturge-Weber associated glaucoma is different than other glaucomas. And so patient aqueous humor could be, again, a really valuable tool to start trying to understand the differences there. The thing about the tissue donations is that they're very precious and very difficult to preserve properly. And so it might seem like, well, many people have given tissue samples over the years. Why do we need new patients to donate? Well, it's almost exactly like when you go to the grocery store. You can't keep things fresh. Sorry, we should.
Yeah,
really
that's a good analogy. It's a great analogy.
Okay.
Yeah.
Well, it's almost like when you're grocery shopping. You wouldn't use your groceries from years ago if you're trying to make a really good meal, and we can't do really good research necessarily with archival old tissue samples that don't necessarily reflect what's going on in patients.
While you were talking about that, it came to mind. It might be helpful for folks to understand why their research dollars that they've invested with the foundation have had such great impact. Because I think in some cases people think, oh, there's just a mouse model, or there's research on humans, but it's important to have a wide array of areas. So like you mentioned about the fish model and a mouse model, and for some people, I know this can be a sensitive issue because they don't want to hurt the little animal. I certainly am not one for that. I love animals, but it is important to test it in another model before we would bring it to humans. So what's the benefit of us as a foundation funding fish models and mouse models?
Yeah, that's a great question. So fish models and mouse models are really critical tools for understanding how a disease works, because you just can't always study humans in the same way. And yeah, there are definitely ethical concerns surrounding animal models, and we have ethical panels to try to make sure that we're doing the research in an appropriate way, where we're not harming the animals unnecessarily. And the reality is that if we want to develop any kind of therapeutic for any human disease, the very first thing that the FDA or any health authority in any other country needs to see is some safety and efficacy. Efficacy meaning how well the drug works. some data from a mouse or a rat,
Mhm.
at least.
Mhm.
And so these fish and mouse models, um, developing these sets us up for eventually being able to register a drug in a human clinical trial. Um, as far as I know, the only Sturge-Weber syndrome clinical trials that have happened so far are for drugs that have been approved for some other disease or indication.
Mhm.
And all of those drugs were tested in an animal model first. So having
Mhm.
animal models that are specific to Sturge-Weber syndrome really allows us to then move forward with new drugs, new therapies that are tailored specifically to Sturge-Weber syndrome.
Mhm. 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 Well, that's a nice segue into the next question. At least I think. Can gene therapy work in Sturge-Weber? Do you need the mouse and the information first before you can do that gene therapy?
Yeah, great question. Gene therapy is a really hot topic, and there's still a lot of things that we don't know about the way that it works. And in the next few years, anything that I say today will be um, almost surely irrelevant. But for now, animal models are potentially very useful for understanding if gene therapy is possible in a human.
Mhm. Mhm.
However, there are many gene therapies that can only be developed and given to a human because of the way it works. It's targeting directly your DNA, and your human DNA is different from a mouse DNA.
Mhm.
Um,
Mhm.
so for Sturge-Weber syndrome, just like repurposing uh, cancer drugs, we will have lots of concerns about safety of gene therapy. But the biggest concern is actually targeting in Sturge-Weber syndrome, targeting the abnormal vasculature, targeting the tissues that are causing the disease, because those tissues are very, very similar to the healthy tissues and very close to the healthy tissues. And so I think it's an open question whether gene therapy can work in Sturge-Weber syndrome, but I think it's worth looking into.
Mhm. Mhm. Well, that's very good news. We hear from folks that are on the internet all the time, and they see all these other disease states. They're going forward with 3D modeling of an organ and all this sci-fi type of things. That very definitely we can get there. I have no doubt that if we think outside the box, and when Dr. Loeb starts talking about the brain vascular malformation consortium, that'll be he can address more of that. But sometimes people get frustrated. It's like, why haven't you done more? I mean, you've been doing this for 35 years. Well, why haven't you done more? And I would assume part of the answer would be funding, and part of the answer would be mouse models and dedicated people. Would you agree with that, or what would you say to minimize those frustrations, I guess?
Funding is always extremely helpful. And I think one of the really valuable things that the Sturge-Weber Foundation has done is establish grants, especially for early stage researchers who want to start studying Sturge-Weber syndrome, and really encouraging scientists who might be studying vascular disease or neurological disease, but are unfamiliar with Sturge-Weber syndrome, to apply for funding to use their specific skills in their laboratory to further progress the understanding of Sturge-Weber syndrome and how it works. So
Mm-hmm.
the funding through the Sturge-Weber Foundation is critical, and we've given out, I think, 2 to 3 grants per year
Mm-hmm. Mm-hmm.
over the last several years. And um, science moves slowly. So clinical research um, moves very slowly because we don't want to harm people. Doctors take the Hippocratic oath and promise to do no harm, and that requires patience and practice.
Mm-hmm.
And for basic science researchers, we also tend to move pretty slowly. We need to understand what we're looking at, and critically, we need people to replicate we're looking at, which means that we mostly have to do things multiple times. So 30 years in the grand scheme of things is actually not a massive amount of time, and we've made a lot of progress in the last 10 years. And we went from not understanding the genetic cause of Sturge-Weber syndrome, having now understood more about the genetics of Sturge-Weber syndrome, opens up, a vast number of possibilities for basic science research. And, in each new finding that comes out about Sturge-Weber syndrome can open up potentially new fields for studying Sturge-Weber syndrome. And
that is a great segue to the next thing that I had. Could you tell them a little bit about how the latest grant that the foundation awarded to the Austrian group is exactly laying that groundwork for what we'll need for the future? And I'm referring obviously to the IPS study. Maybe explain a little bit about what it is and why it's important.
Sure. So one of the grants that we issued this year is for a group that specializes in making induced pluripotent stem cells. And so people have heard about stem cells, certainly. And Sturge-Weber syndrome is really a disease of normal tissues that are just slightly different. And every tissue in your body during development comes from a stem cell that can turn into a blood vessel or a muscle or a neuron. And what we can do now in the laboratory is we can reproduce this developmental process. We can understand developmentally what happens when you make a vascular cell, a cell that's in your blood vessels. And how does that cell look different when you turn it into a neuron? And importantly, if you can make these cells contain the Sturge-Weber syndrome mutation, and if you can grow them both together, you can actually start to make, we were talking about model organisms like mice. You can actually make a system that you can study that's just the cells that you want to try to study together. So vascular cells and neurons, and combine them in ways that you can really control and understand the interplay between the blood vessels and the neurons. And this is not my field of expertise. I'm a computational cancer researcher.
It's a great explanation so far.
Yeah. But the reason why this is critical is because it opens up sort of another possible set of tools for researchers to address safety of different potential therapeutics for Sturge-Weber syndrome, and also systems to understand just how Sturge-Weber syndrome develops in the human body without studying an actual human body.
Mm-hmm. Mm-hmm. Well, I'm very, very excited, especially that the board of directors awarded the $84,000 this year, our largest single year of funding that we've been able to do, because I think it was a nice balance of looking at glaucoma in a different light, and then also preparing for the future with the IPS study to see, how that's going to impact. One of the things I'm most proud of with the foundation is our ability to be able to, again, bring researchers from around the world together, clinical and basic, and they work together really, really well. The clinical care networks across the country, I think it's important because it's local. You may have to travel a few hours if it's not right in Dallas or Houston or Chicago, but you know that you're going to get the best clinical care with the most up-to-date information. And it's the same thing with the International Research Network that Dr. Shirley is leading. I think it's a great collaboration. It's a great way to bring different models together so they can brainstorm about what what's the best approach for a certain type of research, and they play very well together. Let's put it that way. Last question, Dr. Shirley, and we understand you've got some other things to get to today and appreciate your time. What challenges lie ahead, and how do you think we're going to make the best progress with that?
Yeah. Well, I think that the biggest challenge that lies ahead is measurably improving the quality of life for patients living with Sturge-Weber syndrome, right? And so I think that most of that impact comes from the clinical research network and the clinical care network. And so I really don't want to understate how much I think the practical changes to the way that care is provided clinically to Sturge-Weber patients. That's a huge challenge, just coordinating so many different people across the US and across the world. But I think that almost makes the basic research side of things look easy, right? There really are not that many Sturge-Weber labs. And I think that one thing that the Sturge-Weber Foundation has been doing and will continue to do is build sort of a coalition of researchers and encourage collaboration. And that's a challenge that I think we have been really meeting, but it's going to continue. We're having the Sturge-Weber research meeting this July, and really just reaching out and finding new people to bring into the Sturge-Weber research network is one of our big challenges over the next few years. Mm-hmm. And you mentioned potential new therapies, gene therapies that might be developed specifically for Sturge-Weber. I think those are our big research challenges over the next 5 to 10 years, to identify how we can bring new therapies specifically tailored for rare diseases such as Sturge-Weber syndrome to the clinic. And for that, we need to really understand how Sturge-Weber syndrome develops, what it is, and make very useful tools for studying Sturge-Weber syndrome in the lab. And so we're making good progress, encouraging researchers to build these tools and ask these questions. And I think that we need to keep funding that as much as possible.
Funding is key to everything that we do. And I when I first started the foundation, and we started getting into the world of the National Institutes of Health and the FDA, I was amazed. I knew it took money to fund research, but I was amazed at the many different avenues and ways that you can fund it. For instance, the foundation, as you mentioned, gives the research grants. We give seed grants that'll test a little theory. For instance, Naim Issa at the University of Miami only asked for $5,000 to buy some compounds, and now he's on to some greater research with some other collaborators in Boston. So that little seed investment was I think, for us, phenomenal. And then through the NIH, obviously, there are different funding mechanisms that, because we've tested it with that little seed money, you all can go on to apply for greater grants. In fact, Dr. Joyce Bischoff, I believe, and Dr. Nathan Lawson are in a collaboration now that's a multi-million-dollar grant that they've gotten. So it's not that the foundation's members or their family members or loved ones have to fund all the research. It's just that by investing in our research network and in our clinical care network, it gives a strong foundation that then you all can go on to access other avenues of funding that would really open the doors for our understanding. So thank you for making that comment.
Really well said.
Yeah. So I appreciate it. Very eloquent. And if you have any questions for Dr. Shirley, you can email him at cso@sturge-weber.org. And I'm sure he'll be more than happy to answer them for you. Or you can email me to kball@sturgeweber.org, and we'll take care of those questions. So we decided to, have this discussion today in two parts, so you can understand the breadth of what the foundation has put in place, and a nice segue from the funding aspect to the clinical side. Is Dr. Jeff Loeb is joining us today, and a couple questions have been thrown at us that we thought you would like to hot potato. One of the ones was, if my child had neurological symptoms in the past but is stable now, what does the future hold?
Hi, everybody. Well, I think that's the million-dollar question. And it's the question we don't know the answer to. And I think that's what makes a lot of families and people with Sturge-Weber a little scared. What's going to happen? There's something in my brain, kind of looks like what's on my face. It changes over time and can cause a number of symptoms that can be quite scary. And what's going to happen next is really the big question. And that's Karen, thanks you for mentioning our BVMC, our Brain Vascular Malformation Consortium project. This is one of the NIH grants that have kind of followed on from seed funding from the foundation, and also in close cooperation with the foundation. And as many of you have heard, this grant really is to answer those questions. What's going to happen next? We can learn from those who it's already happened to. So it's a longitudinal study to get detailed clinical information, not just what you remember and what your family remembers, but actually go to your medical records, to your caregivers, to fill in a database so that we can actually go all around the country, and if not around the world, and map out what happens in the disease, what does happen next to others, and that may help us predict what's going to happen to you.
Mm-hmm. Mm-hmm. So at this point, we really don't know, because some individuals with Sturge-Weber, they'll have seizures at 6 months of age, and they'll get on medication and they're controlled and life goes on, and they go to elementary school and do sports, and then others, they have seizures at a young age, and they continue to have seizures, and then they have to have half the brain taken out because we can't arrest the progressive issues that are happening. So they're really, at this point, as frustrating as it is, as a parent and a professional, we really can't predict categorically what the natural history is going to be. But that's why the BVMC is so important. And we had discussed earlier in this chat with Dr. Shirley why tissue samples are so important, and the need for again, sharing the natural history amongst one another to learn from it is so important. The BVMC, tell us a little bit about how you're going to bring all the different pieces of the puzzle together. Like, do you need brain scans? Do you need tissue? What all do y'all need to make the BVMC succeed?
So back to your point about how do we make predictions? How do we know who is at the greatest risk to start having seizures? I actually, as an adult neurologist, have Sturge-Weber patients who come see me. They've had no seizures their whole life, and they turn 40 or 50, and all of a sudden they start having seizures. So, we think we know the answers. We think we know what's going on, and then we realize we don't. So we're not sure what those clues will be. Those clues could be coming from the medical records in terms of how old you were, what symptoms you had, what the port-wine stain looks like, whether you have glaucoma or not, and what does the lesion look like in the brain. So we don't necessarily have to get tissue because we have lots and lots of MRIs and CAT scans, and we're collecting all of these, not just at one time point, but over time. So if there's a change in the brain scan that correlates with when people start getting seizures, we can monitor this, see those changes, whether they occur, and then prophylactically prevent or treat the seizures. And that's really the goal, is to identify what's going on in the brain from the scans, what's going on clinically in terms of symptoms related to those scans, because the scans is really representing what we call the pathophysiology. That's That's the substrate of the seizures. It's where it is in the brain, whether there's calcification, where's the angioma, how big is it? All of those things may help us in predicting who is at greatest risk. And it's not just important to let people know what to expect, but it's really important, as Dr. Shirley mentioned, we need some clinical trials. We need to find drugs that work, both from these retrospective studies that we're doing, but also prospectively. We can say, my gosh, look at all the folks who are taking this particular drug. They seem to not have seizures. So let's go do a clinical study now and take everybody who has a brain scan that shows they're at high risk for seizures, randomize those to that drug, and see if it really works. Then we can actually say definitively, aha, let's put people on this medication and they won't have the seizures.
Mm-hmm. Mm-hmm.
So that's what the BVMC is about.
Okay, well, we will definitely get information for everybody so that they know how to participate in the BVMC and through their nearest clinical care network center site. That brings up a nice another segue that I hadn't thought to bring in, but I think it's important that we do. How important is it for Dr. Csaba Juhasz's NIH grant and a study that he's doing where he's scanning adults again that he had scanned when they were younger? How important is it for what he will learn from that study to our understanding of Sturge-Weber?
Well, as you know, Dr. Juhasz's at Wayne State University in Detroit has been a long time, probably a pioneer in brain imaging and Sturge-Weber syndrome. He works closely with us with the BVMC. He's doing all the analysis of the brain scans, and his own projects that are also funded by the National Institutes of Health are really probably, I think, some of the most important clinical studies that we have to understand this idea of pathophysiology, the relationship of what we see on the brain scan to what happens clinically, both in terms of seizures, in terms of stroke-like episodes, in terms of headaches, and really importantly, cognitive and behavioral issues. I remember when we had one of our earlier meetings with the families, and all the doctors and scientists were focusing on the brain and the genes and stuff like that. And everyone, all the families and parents and stuff, they said, well, it's the emotional stuff. It's the impact. It's how this is affecting my life that's most important. And we as clinicians and researchers, we need to constantly keep that in mind, that that's what we're here for, is to really focus on that quality of life. And again, the scariness of what could happen next.
Mm-hmm. Mm-hmm. Definitely. The next question that was submitted was what types of new treatments might be used for Sturge-Weber or the vascular overgrowth? I thought it was a great question because automatically people usually say, well, let's use a pharmaceutical, but there may not be just a pharmaceutical in our arsenal of tools. Are there other things that we might be able to utilize for treatment?
Well, again, that's what we hope to learn from the BVMC. We'll go back and look at hundreds, if not thousands, of patients with Sturge-Weber. We'll see what treatments they had, and we'll use statistics to try to say, well, those who had this treatment actually seem to do better than those who didn't have that treatment. And that'll give us those clues that then we can test, as we say, prospectively in clinical trials. In terms of other treatments, we know that surgery is something that can really help young children who have seizures that are so bad that they're not developing properly. Surgery is still an important, it's not something we really want to take lightly because it's significantly risk. But again, the younger you do these surgeries, the better the outcomes. It is something that still works. In terms of other treatments, I think we have to think outside of the box. We have to look at existing treatments, but we also have to think of new treatments. One thing we're thinking about is the calcification. Everybody sees the calcification, the tram track pattern calcification on the brain. And, oh, that's just something that's a sign. But is it a sign or is it a symptom, a cause? Does it actually cause the symptoms? And Dr. Juhasz's also has some data suggest that those who get the calcifications tend to have worse seizures and stroke-like episodes and headaches. So could we prevent the calcifications from forming? Could we treat them once they're there with water softeners? There's outside of the box things that we can do that we as a foundation are committed to trying these things and really looking for something that makes a difference in people's lives.
Mm-hmm. Well, we're so blessed to have both of you committed, and especially you, Dr. Loeb, for such a long time to our rare disease. I'm excited because we've laid the groundwork with the SWFern, and we've laid the groundwork with the CCN and the foundation's new initiative. We've been reaching out over the past year to all the other organizations around the world, France, Germany, Italy, and working to build a coalition and a council where we can bring all of our clinical and basic researchers within each country to come together to pool their resources, their knowledge base, and expertise, and then to also work with the families of these other organizations to determine what are the greatest impacts of quality of life in their particular country. What can we share with each other to improve that quality of life and care? So I'm very excited for 2022. That initiative will move forward. We'll have a meeting with them, and I see we only have anywhere to go but up. So thank you
No,
both
I
so
think
much.
that's very exciting. I'm so happy to be here and part of this. I guess I kind of fell in love with not just the disease, but the people who have the disease and their families, and it's hard to let go once you kind of fall in love. And I'm thinking many exciting things on the horizon. I think the international collaborations are exciting. The BVMC is really about big data. Big data has no boundaries, does not care in the US or in Germany or in Russia or wherever you are. We can build that database and come up with these treatments together across the world.
Very much so. Well, thank you so much. Is there anything else either one of you would like to bring to the discussion or encourage others to do?
I mean, I'd just like to, again, encourage you to reach out to any of our sites for the Brain Vascular Malformation Consortium, BVMC. It takes some work. You've got to collect the medical records. You've got to collect those images. But I think by everybody participating, you're not only helping your own family and family members or yourselves who have the disease, but others around to learn from your experience so that they can have hopefully a better experience going through life with this rare disease.
Very much so. Thank you so much. We look forward to round 2 of this discussion and appreciate y'all participating today.